Hello, I'm Dr. Aizaz from MedicoVisual.com, and in today's visual lecture we will talk about the development of Diencephalon. So previously we had already discussed that the neural tube forms different swellings, right? So we had discussed that from the neural tube different swellings develop, and if we talk about those swellings, initially, three primary brain swellings or primary brain vesicles develop, and these are prosencephalon, the outermost, the cranial one, I should say. This is the cranial most. It is the prosencephalon. Then we have this one is this was the prosencephalon. Then this is middle one. This is the mesencephalon, and the last caudal most one is rhombencephalon, and of course finally here this part is going to form the spinal cord. Now this lecture is also a bit of revision so that you get the concept here. Now, in the lecture of rhombencephalon, we had discussed that how the roof plate in this rhombencephalon region, it's a place open. Here you see it's a place open and it widens up like this, right? So we had discussed these things. Of course, the rhombencephalon region, it will be divided into two parts, and here this prosencephalon region, the neural tube, not neural tube, I should say neural canal, the inner one, this one is the neural canal, this one is the neural canal. This neural canal, it will sort of dilate, and you see what is happening here. And by the way, here the rhombencephalon, it is divided into two parts. So rhombencephalon has been divided into the upper or cranial part, which is metencephalon, which is going to form pons and cerebellum, and the caudal part is myelencephalon, which is going to form the medulla oblongata. Of course, in the dorsal region of the metencephalon, ventral region has formed the pons, but the dorsal region is going to form the cerebellum, right? So here you see there is that dorsal lip, the lip of rhombic lip basically, and from that rhombic lip the cerebellum develops. We had discussed these things already. Now here our focus will be in the prosencephalon region. Now initially you see that there is a single swelling of prosencephalon. Again, let me reiterate these things. This might confuse some of you that this whole swelling here, this is currently the prosencephalon. Then this is mesencephalon, and this both of these swellings which have now formed three structures, the pons, medulla and cerebellum. This is rhombencephalon, and rhombencephalon, of course, it is divided into meten and myelencephalon. Now what will happen in the prosencephalon region is that here an outgrowth develops in the upper and slightly dorsal part of prosencephalon. This prosencephalon, here the lateral outgrowth of the neural canal develop, and of course along with neural canal the other neuroblast, they will also proliferate, and then the prosencephalon will be divided into two parts. Let's see that. So you see outgrowth develop, and then it has divided into two parts like this. So now the prosencephalon has been divided into two parts. The inner one, here is the inner one. The inner part is diencephalon. This is the inner part. This is the diencephalon. Let's use this color. This is the diencephalon, and the outer part, this is the outer part. This is what? This is the telencephalon. So inner one is diencephalon and outer one is telencephalon. So you see that diencephalon is surrounded. It is surrounded from outside by the telencephalon. If I draw the diagram here, you see this is the diencephalon, of course mesencephalon and rhombencephalon, right? Now here you see this connection, and then like this is a situation that you see such type of diagram in the textbooks, that it is, you see this one is the This is the telencephalon, and this one is the diencephalon. Now if you focus on the central canal, you see that in the spinal cord region it has formed the central canal of the spinal cord. In the rhombencephalon region it has formed the fourth ventricle. Then in the mesencephalon it is still narrow, and here it has formed the central aqueduct of Sylvius of the mesencephalon or midbrain. Then it dilates in the diencephalon region. It dilates in the diencephalon region to form which ventricle? Third ventricle, right? And finally here it is forming in the telencephalon region. It is forming this relatively large ventricle. You see, huge ventricle. This is the lateral ventricle. These are basically bilateral. This is, for example, first ventricle. This is second. Actually, it is not clear whether we should call the right one as first or left one as first, so arbitrarily I am writing these first and second. These are basically lateral ventricle. So if we consider the lateral ventricle first and second, then this is the third ventricle. That is why it is called third ventricle, right? Then this is the central aqueduct of Sylvius, and then this is what? This is fourth ventricle. And this is not fifth ventricle, actually. This is the central canal of spinal cord. So this is the situation. And by the way, you see that third ventricle is connected to first and second, I mean the lateral ventricle, with the help of this connection called foramen of Monro. You see this? Let me show you again. This one, exactly to be precise, this is the interventricular. I mean, it is between the ventricles. It is connecting the ventricles. So ventricular foramen of Monro. Now the structures that are surrounding the third ventricle, these are basically diencephalon, and structures that are surrounding the lateral ventricles, these are basically the telencephalon derivatives, right? So telencephalon derivatives are mainly the cerebral cortex and basal ganglia that we will discuss in the next lecture. In this lecture we will focus on the diencephalon, and diencephalon derivatives are in this region, in the region of third ventricle. They are surrounding the third ventricle, or if we put it in another way, we can say that the third ventricle is the cavity of diencephalon, and lateral ventricles, they are the cavities of telencephalon. And in this way we can continue that, we can extrapolate this discussion that the cavity of mesencephalon is central aqueduct of Sylvius, the cavity of rhombencephalon is third ventricle, the cavity of spinal cord is central canal of spinal cord. So in this way, the ventricular system as well as the neural system, the neuronal structures, they both are developing side by side. And I hope that you understood that the diencephalon, it is deeply seated. You see, diencephalon, it is deeply seated here. The diencephalon is deeply seated within it is surrounded by the telencephalon. You see, it is surrounded by telencephalon. So now our discussion will be mainly related to diencephalon in this lecture. Now we will focus more on diencephalon after this basic overview. Now we will focus on diencephalon, and now we will discuss that how the structures related with diencephalon, how do they develop, and we will see what are those structures as well in a bit more detail. So now here you see all these structures as we have discussed previously. That blue one here is the alar plate. Basically, the neuroblasts in the neural tube, they reorient themselves to form these aggregates of neuroblasts in two regions. The dorsally placed are the alar plate, and ventrally placed are the basal plate. So these red one here, these are the basal plates. Basal plate, we have discussed these things previously as well, and dorsally placed, these are the alar plate. Now these structures, they are going to form the gray matter of different parts of the central nervous system. We had discussed how they form the different components in the spinal cord. We have also discussed their functions, their derivatives in rhombencephalon and mesencephalon. Now again our focus will be only the diencephalon region. So here this is this especially this part. Now here this part is the diencephalon. Up to here this is the diencephalon, and below that is mesencephalon. Now if we focus on diencephalon, what actually happens that here the basal plate is actually regressed. Some authors believe that basal plate do not develop in this region. Other authorities related with embryology, they believe that although basal plate develop, but basal plate is mostly regressed. Almost all of the basal plate in this region is destroyed, it is lost. So what actually happens that, so you see basal plate is being destroyed. So what actually happens that only the alar plate remains in the diencephalon region, and alar plate is going to form the structures in the diencephalon region. So this area, this central canal of diencephalon region, it will dilate, it will grow, and of course along with that alar plate will also proliferate. And now this alar plate, it will completely surround this dilated part. This dilated part is, by the way, now called third ventricle. So now alar plate, it will, the cells in the alar plate, the neuroblasts in the alar plate, they will proliferate and they will almost completely surround this third ventricle like this. So they have completely surrounded the third ventricle, and for the sake of simplicity and ease of understanding, I have removed the other parts. You only see the third ventricle of diencephalon and some part of central aqueduct of Sylvius, which is basically the part of the mesencephalon or midbrain, right? So it has surrounded, the alar plate has completely surrounded this, and this alar plate is going to form all the structures in this region. So now what will happen that alar plate will be divided into different parts. Alar plate of diencephalon will be divided into different sections. So you see this section, these are the sections here, and let me tell you what are these, and then we will understand what that is going to do. So this near the roof plate, this one, this is the epithalamus. This is the epithalamus, or you can say this is primordial epithalamus. Of course it is not the definitive epithalamus, this is primordial epithalamus. This is the part of alar plate which is going to form the epithalamus, right? Then this relatively bigger part, this is the primordium of thalamus. And this part which is in the floor plate, this is, by the way, here there will be the floor, there was the floor plate. So this is the primordium of hypothalamus, and here this small part is the primordium of subthalamus, right? So now these structures are going to form the epithalamus, the thalamus, subthalamus and hypothalamus. And if I show you here that here there was the roof plate, here there was the floor plate, and you see the situation here exactly. And it's very easy to understand that ultimately that these alar plate derivative structures, they are going to form the structures that I've told you. The thalamus here, this bigger one, thalamus, the subthalamus, here is the epithalamus, and here is the hypothalamus. Now actually these two things, these are basically called habenula. I know it looks like a name of some kind of monster or alien. So basically these are called habenula or habenular nuclei. And these are interconnected with each other through the structure called habenular habenular commissure. Actually anything, not anything, basically it's a collection of nerve fibers, or you can say it's a white matter tract. So basically a tract which connects the bilateral structures in the central nervous system, they are called commissure. So these two habenuli, they are connected with the help of this habenular commissure. And here is, by the way, posterior commissure. I will tell you what it is in a moment. This is the posterior commissure. And the interesting thing here, you see, it's not the adult stage, of course it's the embryonic stage, and at this embryonic stage you see that here is this thickening of epithelium in this region. In this epithalamus region, there is this thickening of epithelium here in the roof plate region. You see, this is the roof plate. So in the roof plate region, this is a thickened epithelium here. This is a thickened epithelium here. Now what will happen, that this thickened epithelium will evaginate. What I mean by evagination is that initially it is like this, but it will grow outward like this. You see, it was first straight, but it has grown outward like this. So this is called evagination. So here this thickened epithelium of the epithalamus region, it will evaginate, and it is going to form what? It is going to form the pineal gland. So now these three structures, not three, one, two, three, four, five. So these five structures as a whole, they are considered as the part of epithalamus, or all these structures you can say these are forming the epithalamus. What are these structures? Right and left habenular nuclei or habenula, then habenular commissure, third, then we have pineal gland, fourth, and finally fifth one is the posterior commissure. Now this posterior commissure is actually connecting the pretectal nuclei together, and some authors, they also believe that they are also connecting mainly they are connecting pretectal nucleus, posterior commissure. They are interconnecting the right and left pretectal nucleus. That is most important thing, please remember that. Along with pretectal nuclei, along with pretectal nuclei, some authors also believe that they are connecting right and left superior colliculi, and some authors, they also believe that they are interconnecting right and left medial longitudinal fasciculus. What are these? We will discuss that later, and not in this lecture series, in the neurophysiology and neuroanatomy lecture series. But regarding the pretectal nucleus and regarding the importance of these posterior commissure, I will tell you a few words a bit later, right? So all these structures are basically the epithalamus, right? Now if we come forward, this is the hypothalamus, and this is subthalamus and thalamus. Now what will happen there, these right and left thalami, they will grow larger, and as they grow larger, subthalamus is below the thalamus. So subthalamus will become slightly laterally, you see, subthalamus will become slightly laterally like this. And second thing is that the third ventricle, its structure will also change, it will become biconvex, right? And third thing is that the thalamus, as they become close together, they are opposed to each other, so they will become interconnected with each other. Let me show you. Let's isolate the thalamus. You see, now they are interconnected with each other, and this is called intrathalamic adhesions. So there are adhesions between the two thalami, so interthalamic adhesion, right? So, and if I show you the third ventricle, you see it has become more like this biconvex. You see, due to presence of these huge thalami, it has become biconvex like this. And in this third ventricle, you see this hole that allows this interthalamic adhesion, right? So thalami are interconnected with each other. And what are these two things here? Bilaterally, you see, These are geniculate bodies again, which also develop from the diencephalon, from the alar plate of diencephalon, specifically from the thalamic region, primordium of thalamus. So this is lateral geniculate body, which is related with vision. It is the component of visual pathway, and this is slightly posteriorly, and as it is posteriorly, of course, it is more closer to midline. And this is less closer, it is a bit far from the midline. So it is lateral geniculate body, and it is the medial geniculate body. Personally, I believe that the lateral geniculate body should be called ventral geniculate body, or anterior geniculate body, and this medial geniculate body, it should be called posterior geniculate body or dorsal geniculate body. That would be more logical naming scheme, but this is how they call it in all of the textbooks. That is the naming scheme that is being followed, so you will have to follow the same. We cannot unfortunately come up with our own naming scheme, right? Finally, a few words about the hypothalamus, the pink one here, that here in this region, in the inferior region of hypothalamus, two swellings developed, and these are called mammillary bodies. So you see these two breast-like swellings develop here. These are called mammillary bodies. Later we will see that infundibulum also develops here, and then the pituitary gland develops here. Development of pituitary gland will be discussed in the next lecture. So you see this huge thalamus, subthalamus, hypothalamus, and the epithalamus. So I think that is all about diencephalon. But before ending this lecture, I will discuss a few things related with the visual pathway, right? Now actually, the diencephalon, not only these structures are that are derived from diencephalon. Actually, even before the diencephalon properly develops, even at the neural tube stage, the region which is going to later form the diencephalon, and later which is going to form lateral geniculate body of the thalamus, in that region, of course, at that stage there was no lateral geniculate body, but the region that is later going to form the lateral geniculate body, from that region these extensions develop like this, these evaginations develop like this, and these are going to form here. What is this? Here they will cross like this, and this is called optic chiasma. These are optic tracts basically. These are going to form optic tract. Here near the hypothalamus, of course, here will be what? Pituitary gland. So near the pituitary gland and hypothalamus, here this is the optic chiasma. So optic tract, this is optic tract, and here is the optic chiasma. Later it will grow further actually, and here it is going to form optic nerve, and this optic nerve will actually then go further, and here the retina will be formed from this, and some part of iris as well. So optic cup, and we have discussed actually these details in our lecture on the eye development. You can see it further in proper 3D. I am just giving you a review of these, that you can correlate this thing with that eye embryology as well. You don't forget that. Now all these structures, the retina, some part of iris, some part of ciliary body, the optic nerve, the optic tract, optic chiasma, and of course the other things that I've discussed already, that thalamus, lateral geniculate body, epithalamus, hypothalamus, all these structures, they develop from diencephalon. So please don't forget that it is not the thalamus and related structure. Actually the retina and other eye related structures, they also develop from the diencephalon, and I have discussed their complete details in eye embryology lectures. You can watch that as well. So I just wanted to correlate these things. Secondly, we had discussed the pretectal nucleus. You know what is the function of pretectal nucleus? We have already seen that some fibers. So if let's suppose here is the pretectal nucleus, it is slightly above the midbrain. You see below that is midbrain, of course. Below that is midbrain. You remember that? So this is crus cerebri, and of course this is superior colliculus. Below that will be inferior colliculus. I hope you understand these things. Of course there will be pons here. So anyhow, you got the concept. Now here these are the pretectal nucleus. So now pretectal nuclei, basically pretectal nuclei, they are getting the light information from the optic tract here, and then pretectal nuclei, they are going to send the information. They are going to send the information to the Edinger-Westphal nucleus here, and from Edinger-Westphal nucleus through the third nerve, through the third cranial nerve, information will go to through the long ciliary and short ciliary nerves. We will discuss these things later, and ultimately they will go to iris, right? And they are going to, this is the basically not exactly iris, the constrictor pupillae muscle. So here they will go to constrictor pupillae muscle. Now here you see that pretectal nuclei, they are connected together. This is right and left pretectal nuclei. They are connected together through the posterior commissure. We have discussed these things. Now for example, even if I shine a light on this eye, this is let's suppose right eye. So if I am going to shine the light on right eye, what will happen that the pupil of right as well as left eye both will constrict. You can try this pupillary light reflex. You can shine the light on a person's right eye and you can see that even the left eye's pupil will constrict. How it happens? That information is going through the optic nerve to the optic chiasma, optic tract, and ultimately it will go to pretectal nuclei. Now this is right eye, so you see the pathway here going to here, and here. Now you see what is happening, that here these pretectal nuclei are interconnected with the help of posterior commissure. So because they are interconnected with the help of posterior commissure, these pretectal nuclei, they can intercommunicate with each other, and they will tell that some light is being shown on this eye. So they will tell this pretectal nuclei, they will also stimulate this pretectal nuclei, and so the light information will go here. The parasympathetic information from the Edinger-Westphal, they will stimulate this Edinger-Westphal nucleus. Parasympathetic fibers will fire through this third cranial nerve and go to constrictor pupillae of other eye as well. Of course they will also go to this eye, but they will also go to other eye, and they will also constrict the other eye as well. So that is the significance of posterior commissure. And of course don't forget that some authors, they also believe that, of course this is the superior colliculus. Some authors, they also believe that posterior commissure is also interconnecting the superior colliculi. So I think that was it. Finally, if you want me to review these things, we can draw a flow chart here. That first of all, there was the neural tube. You can skip this part if you already understood it properly, but if you want to have a quick review, you can say that neural tube is divided into prosencephalon, mesencephalon, and rhombencephalon, and of course spinal cord as well. Now forget about other structures. If we discuss specifically about prosencephalon, it is divided into telencephalon and diencephalon. Now from diencephalon, what will happen? First of all, there is the eye related structures that we have discussed already. The optic tract, the optic nerve, optic chiasma, retina, iris, and part of ciliary body, and all these structures. So this is eye related. Then the diencephalon is divided into non-eye related structures. And then what are non-eye related? This we can divide into the big one, thalamus. It is, by the way, also called dorsal thalamus. Some people also call it dorsal thalamus. I don't know why, to be honest. Then we have what is this? Epithalamus, right? Epithalamus. Then we have hypothalamus. Now from the epithalamus we have discussed that there is habenula and habenula plus commissure plus habenular commissure. Then we have pineal gland, and then we have posterior commissure. And by the way, some authorities, they also believe that pretectal nucleus, it does not develop from the alar plate of midbrain, it actually develops from alar plate of thalamus, specifically most likely developed from the alar plate of epithalamus. So we can also include, this is not confirmed, but we can also include the pretectal nucleus here. Pretectal nucleus. So hypothalamus, it will develop into hypothalamus. Along with that, hypothalamic nuclei, of course, almost all of the hypothalamic nuclei, not almost, actually all of the hypothalamic nuclei. And along with that, in next lecture we will also discuss that it also form posterior pituitary, which is actually not a true gland, it is actually an extension of hypothalamus, that we will discuss in the next lecture. Then dorsal thalamus, it is going to form this main bulk of thalamus, which includes several thalamic nuclei that I am not going to discuss in this lecture, that is a huge topic. So it is going to form all the thalamic nuclei, and it is going to form metathalamus as well. Metathalamus. Now what is metathalamus? Actually, it's nothing more than these lateral geniculate body and medial geniculate body. So in some textbooks you might have seen written metathalamus. Metathalamus is basically medial and lateral geniculate body. These two structures, they are combined as a whole, they are also called metathalamus, right? So this is the whole of the crux of this lecture. And of course, please don't forget that almost all of these structures, they develop from the alar plate. Basal plate is regressed in this region. So that was all about development of diencephalon. In this lecture, I will not go into functions of these structures because this is very complicated and it's not possible to describe all these functions. But okay, let me tell you one or two lines about each of this if you want. Otherwise, you can skip this part of lecture. Now, out of this epithalamus, we have discussed actually the functions of the pretectal, this commissure, posterior commissure. Regarding habenula, I will not go into details. Regarding the pineal gland, it is one of the most important structures in this region, and it is related with circadian rhythm, the day and night rhythm. Actually, it modulates our nervous system and endocrine system. Specifically, it modulates the autonomic nervous system as well as our endocrine system as per the light condition, as per the day and night timing. It is actually getting the light information through hypothalamus. There is a long pathway that I will not go into detail, and it actually releases a chemical called melatonin, and that melatonin, it is related with circadian rhythm. Then this hypothalamus, it integrates all the sensory information. The lateral and medial geniculate body, we have already discussed that lateral geniculate body is related with vision, and medial geniculate body, it is related with auditory pathway. This is related with visual pathway, lateral geniculate body, and medial geniculate body is related with visual, the auditory pathway. Regarding the subthalamus, it is actually the functional part of basal ganglia that we will discuss in the upcoming lectures. By the functional part, I mean that most authors, they do not include it as a part of as a part of basal ganglia. Historically, it has not been included in the basal ganglia, but functionally it is still considered as a part of basal ganglia because it has a lots of connections with the basal ganglia. And then this hypothalamus, this is one of the most important part of your body. It's a very small, its weight is less than a fingernail, but it controls all of the functions of your body, the autonomic functions, the endocrine functions, and a whole 2-3 hours lectures can be dedicated on the functions of hypothalamus. This is very important structure. So I think that is all about the development of diencephalon. I hope you learned something from this video. In next lecture we will discuss the development of pituitary gland, and that is very important topic, to be honest. So thank you so much for watching this video. Have a great day. Goodbye.